Some proteins undergo assisted folding. Not all proteins fold spontaneously as they are synthesized in the cell. It means कुछ प्रोटीन्स ऐसे होते हैं जो कि स्पॉन्टेनियसली सिंथेसाइज नहीं होते हैं सेल में, मतलब कि सारे के सारे प्रोटीन्स उसी तरीके से फोल्ड नहीं होते हैं. Foldings for many protein is facilitated by the action of specialized protein that is molecule chaperones. अब हमने अभी तक एक कॉन्सेप्ट पढ़ा था कि जो प्रोटीन्स होते हैं उनमें एक टेंडेंसी होती है कि उनमें एक ऐसी मेमोरी होती है कि वो हिडन ट्रायल मेथड नहीं करते. उन्हें पता होता है कि उनको कहाँ पे फोल्ड होना है और वो वहीं पे जाके फोल्ड होते हैं, जिसकी हेल्प से they used to make that folding of protein. But while talking about some few proteins which do not undergo folding by spontaneity, so we have some molecule chaperones which used to help that protein to interact and to fold. So these used to give an partial folded or improperly folded polypeptides, facilitate correct folding pathway or providing microenvironments in which folding can occur. तो ये ऐसे फोल्डिंग पाथवेज प्रोवाइड करते हैं या फिर माइक्रो इन्वायरमेंट्स प्रोवाइड करते हैं सो दैट फोल्डिंग कैन बी easily done. Two classes of molecular chaperones have been well studied. So might be possible there are more than two classes of molecular chaperones, but we have studied two classes very well. Both are found in organism ranking from bacteria to humans. सो बैक्टीरिया सबसे ज़्यादा प्रिमिटिव होते हैं. अब हम पहले चैप्टर को अगर रिवाइज करें तो पहले चैप्टर में इट इज़ रिटर्न दैट यू बैक्टीरियाज विच आर और आर्की बैक्टीरियाज विच आर फर्स्ट लिविंग बैक्टीरिया, तो उनमें भी ये प्रेजेंट होता है. एंड ह्यूमंस विच आर वेरी नाउ इवॉल्व्ड, उनमें भी ये प्रेजेंट होता है. द फर्स्ट क्लास अ फैमिली ऑफ प्रोटीन कॉल्ड HSP70 generally have a molecular weight near 70,000 and more abundant in cell stressed by elevated temperature. So we have this HSP70 class which is whose molecular weight is 70,000 और ये बहुत ज़्यादा प्रेजेंट होती है उस सेल में जो कि स्ट्रेस्ड होती है, जिसमें ज़्यादा टेम्परेचर एलिवेट कर देते हैं हम, जिसमें टेम्परेचर. हेंस हीट शॉक प्रोटीन MR70,000 और HSP70. तो बेसिकली ये है हीट शॉक प्रोटीन जो कि अगर टेम्परेचर बढ़ेगा तो उसके साथ इट यूज टू इंक्रीज. HSP70 protein bind to regions of unfolded polypeptide that are rich in hydrophobic residues, preventing inappropriate aggregation. Hydrophobic means those who resist water. So if HSP70 protein binds with those regions of polypeptide, for example hydrophobic has a tendency to make aggregation, so that if this is hydrophilic, so this portion is hydrophobic. So it used to prevent this aggregation by binding to them. These chaperones thus protect proteins that have been denatured by heat and peptides that are being synthesized. So the work of these chaperones here is to protect those protein who are denatured by heat. HSP70 protein also block the folding of certain proteins that must remain unfolded until they have been translocated across membrane. So HSP70 proteins also they used to block the folding of certain proteins. Block the folding means it used to prevent certain proteins from unfolding. Means if they are folded like this, it will not let them to unfold like this. So they used to prevent them from unfolding means while they are dislocating from a membrane, they should be unfolded. मतलब खुले रहने चाहिए, फोल्ड नहीं होने चाहिए. while they are translocating means एक सेल से दूसरी सेल मेम्ब्रेन पे जा रहे हैं. Some chaperones also facilitate the quaternary assembly of oligomeric protein. So कुछ ऐसे भी chaperones होते हैं which helps or which what is quaternary assembly oligomers having more chains basically. The HSP70 protein binds to and release polypeptide in a cycle that also involves several other proteins. Okay, so HSP70 protein, it used to bind to and release few polypeptides in a cycle that also involves several other proteins and ATP hydrolysis. Now we have illustrated chaperones assisted folding as elucidated for the chaperones DNK and DNAJ in E. coli. Now we should know what are the functions of these two. For example, while talking about DNK, which is this, so the DNK interact with both the backbone and side chain of a polypeptide, and it has an polarity which admits only L peptide segment. While talking about DNAJ binds with both L and D peptide and is assumed to interact only with the side chains of the substrate. So DNAJ, it used to bind with the substrate side chain. So DNK and DNJ were first identified as protein required for in vitro replication of certain viral DNA molecules. So there were certain viral, those who are present in viruses present, bacteria, those used to have. So यहाँ पे we have shown that thing कि DNJ bind unfolded part. So here we have unfolded protein. This is an unfolded protein. So we have DNAJ here present, and this DNAJ is getting attached to here and here, the ends of the unfolded protein, and what it is making, it is binding tightly to unfold protein, and then it is converting into this. In bacteria, the nucleotide exchange from GRPE stimulates and which releases ADP. Then moving on, here ATP is present in this, and after that ATP binds with DNK protein, and then the protein deassociates, and then after this we have unfolded protein which is present here, and those protein which are not folded properly are then undergo the same process again. So the second class is a little complex and it requires folding of a number of cellular proteins. So for do not E. coli estimate 10% and 15% cellular protein require the resident chaperonin system called GROEL and whatsoever it is written here. These proteins first become known when they were found to be necessary for the growth of certain bacterial virus. So we have written the theory of this protein which you can read, uh, and this is something which is your homework. Now finally we have this, I don't think it is that important, but the cycle is given here. Okay, now moving towards the summary, which you will read it by yourself, because it is also important that you do certain homeworks, like each and everything should not be spoon feeded to you. Now we have given certain problems as well, which I have shown you guys. You should try this. And that's how we completed our fourth chapter. And the fifth chapter is protein function.